Hi, I'm Dr. Hill. I'm here with you today to review experimental design. This is the process of planning a study to meet specific objectives. It's important to plan the experiment because you want to follow established scientific protocols, which will help ensure that the right type and amount of data are collected. Experimental design starts after you have asked a question and formed a hypothesis. In other words, you already know what you want to test. The start of this process is to identify your groups. Good experiments only have two groups, a control group and an experimental group. We'll start with the control group. The control group is used for comparison to the experimental group. If I decided to test to see if tomatoes grew best under natural light or blue wavelength light only, then I would be testing the type of light. Therefore, the natural light would serve as my control because that's what's found in nature. The experimental group is the same as the control group, except that it varies by one variable. In my example, the experimental group would be those tomatoes grown under blue light. Another feature of a good experiment is that each group should have a minimum of 3 replicates. A replicate is an exact copy of a sample that is being analyzed on which the same procedure is done. So in our project, we would need at least 3 tomato plants under both types of light. However, the more replicates that you can produce, the better the data. Now that we have identified our groups, we need to discuss variables. A variable is any factor, trait, or condition that can be controlled or changed. There are 3 types of variables that we will use in experimental design: independent, dependent, and control. The independent variable is the thing that is changed between the two groups. In our project, it would be the type of light. The dependent variable is what is being measured. In our experiment, this would be how much the plants grow. However, you will need to determine how you will measure plant growth. Will this be measured by the height of the plant, the number of leaves on the plant, the number of flowers produced, the mass of the plant, or something else? Control variables are critical to a well-run experiment. These are the factors that are kept exactly the same between groups so they don't affect the results. There should be only one difference between the groups, and that is whatever you're testing. Everything else should stay the same. When planning your controls, it is necessary to have a negative control group and a positive control group. The negative group is one in which no treatment is given. A positive control group is one that receives a treatment with a known outcome, but not the experimental. This presentation was brought to you by the Noble Research Institute.